of the Gene Transfer at Hannover Medical School and the current president of the ESGCT. The ESGCT, or European Society of Gene and Cell Therapy, is a non-profit organization promoting fundamental and clinical research in gene therapy, in cell therapy, and vaccine development. Creating a platform for scientific networking and exchange, as well as education, are parts of our mission. We therefore launched the ESGCT E-Series, a seminar series. The first 3 seminars are dedicated to genome editing and are sponsored by Therapeutics. Last week, as you might have seen, we started with a seminar of Professor Vijay Sitchis of the Institute of Biotechnology of the Vilnius University. He introduced us into the topic and shared impressive new results obtained by his group. Today, the focus is set on safety consideration. This important topic will be presented by Professor Toni Cathomen of the University Hospital Freiburg. He's director of the Institute of Transfusion Medicine and Gene Therapy and internationally highly recognized expert on designer nucleases, including the CRISPR-Cas technology. Toni studied biology in Zurich before joining the Salk Institute as senior research associate. In 2009, Professor Cathomen accepted a professorship at the Charité in Berlin. After that, moved to Hannover Medical School and is, as I already mentioned, since 2012, director of the Institute of Transfusion Medicine and Gene Therapy. Professor Cathomen is very engaged in the field of gene and cell therapy, as indicated, for example, by his current function as president of the German Society of Gene Therapy. Thus, we are looking very much forward to his lecture, which is entitled Safety Consideration in Therapeutic Genome Editing.
Okay, thank you very much for this kind introduction, Hildegard. Try to get my, yes, now it's moving. I think Hildegard already introduced the topic very nicely. I think you heard, everyone who tuned in last week heard is giving a very nice introduction into how CRISPR-Cas has been discovered and maybe also new tools that come out in the future. And my topic today will really be on safety considerations if you apply genome editing tools in a therapeutic content. So I think I don't have to, let's say, repeat to this audience that Emmanuel Charpentier and Jennifer Doudna were awarded with the Nobel Prize last year for their discovery of CRISPR-Cas nucleases. But I think what I would like to point out is how quickly this technology was translated into the clinic. If you think about that, the discovery was made in 2012, and we already have first patients benefiting from cures based on CRISPR-Cas nucleases. I think this is really astounding. So I think I just would like to say that, of course, CRISPR-Cas nucleases are not the only designer nucleases which have been applied in the clinic. And you can see there are tail nucleases as well as zinc finger nucleases. And I think overall, the basic concept of all these nucleases is the same. I think you have the DNA binding domain, which guides the nuclease to its target site, and then you have a cleavage domain, which then introduces the double-strand break. So I think this is common for all these nucleases. I think what you know as well is, I think, the exception, or how CRISPR-Cas is distinguished from these two guys here, is that the DNA recognition domain here is not protein-based, but it's a short guide RNA, which then through Watson-Crick base pairing guides the Cas9 protein to the target site. But I think, as I said before, I think it's really surprising to see how fast these nucleases have been translated to the clinic. When you think about, while zinc finger nucleases have been around for quite a while, the first clinical trials was launched in 2011. And you see that overall more than 50 clinical trials are ongoing. And you will hear probably next week much more about ongoing clinical trials, also from a perspective of a biotech company. And you can also see on this very short list that CRISPR-Cas nucleases, TALENCE, as well as zinc finger nucleases are applied in vitro, ex vivo, as well as in vivo. I think that's also important to know. So maybe just to to quickly introduce what we are interested in Freiburg, and you see this here. These are the platforms that we work with and that we use to develop innovative cell and gene therapies. So we do disease modeling in vitro using iPS cells, for instance, but also in vivo using small animal models. We have a platform which we try to optimize further and further for genome editing as well as epigenome editing. And we also do GMP development, so really to bring these therapies to the clinic. And I think what is very important for me in that context is to really stress that from the beginning, we actually talk to the clinicians and make sure that we understand the problem that they have and develop our products together with them. And we have our clinicians, of course, in the Center for Chronic Immunodeficiency. If we try to develop, let's say, therapies for primary immunodeficiencies, as well as for HIV infection, or in the Comprehensive Cancer Center in Freiburg for our CAR T-cell program. You might think that this is a very broad spectrum, but I think if we really boil it down to the technologies and to the cells, you will see that we actually work with two cell types in the lab, which are the T cells on the one hand and the blood stem cells, the CD34 cells on the other hand. And in terms of technology that we translate to the clinic are really tail nucleases as well as CRISPR-Cas nucleases. And I think with these platforms here, we try to address these disease entities and try to develop genome editing therapies for these patients. I think what I'm going to talk today is not, let's say, what we do in research in terms of new therapeutics that we develop, but I think rather focus on what is important as well. So these safety considerations to go into the clinic. And of course, one safety consideration is how specific are the nucleus that you would like to bring to the clinic. So we developed novel assays to look into specificity of designer nucleases. And the second point which we focus on is the GMP development part. And it's really, again, safety considerations. How can you produce a gene editing product in a safe manner to make sure that when you transfuse it to the patient, that it's still safe and not contaminated, for instance, with bacteria. And I would like to start on this side here, so with the manufacturing problem. And just to introduce you why we are interested in manufacturing, I think I would just like to give a one-slide summary of our efforts to produce CAR T cells that target PSMA, so prostate-specific membrane antigen, to produce a product which can be used against prostate cancer. So we made novel CARs to target PSMA, and we tested them first in vitro in the context of CAR T cells, of course, and we can show that our CAR T cells that we produce are highly effective in killing PSMA-positive tumor cells. We also show when we look at the immunophenotype of these CAR T cells, depending on what co-stimulatory domains we use, we have either more effector cells or more memory cells. And if we then apply these CAR T cells in vivo, and this is just one application that I'll show you here, where we combine or where we infuse the CAR T cells in combination with a chemotherapy to actually, let's say, break up the tumor microenvironment so that the CAR T cells can access the tumor. You can see this here. If we treat these mice, which now bear the tumor here, with chemotherapy alone, nothing happens. If we treat them with CAR T cells alone, nothing happens. But if you combine the two therapies, so chemotherapy and CAR T cell therapy, you can see that we can efficiently eradicate the tumor in these mice. So this sets the stage. So to understand why we're interested in producing CAR T cells. And I mentioned before, I think it's important to develop tools or manufacturing procedures that allow you to, let's say, manufacture a lot of CAR T cells at the same time, and to make sure that you don't mix up, let's say, the cells that you produce for one particular patient and so on. And we teamed up with a company called Miltenyi. And Miltenyi has developed this very nice machine that you see here on the left side. And this machine actually allows us to produce CAR T cells, but also blood stem cells-based product in an automated and GMP compliant fashion. And probably most of you have never seen such a machine before, so just give me a little bit of introduction, because I think it's quite interesting to see how such a, let's say, more expensive machine can work. So here would be the patient material. This can be a ferrisate containing blood stem cells, but also T cells. And in a first step, you actually try to now clean up your cell product. So you can either enrich for CD34 cells, which are the stem cells, or for CD4, CD8 cells, the T cells. So this is done here with this magnet here. And then the cells are pumped to this side here. They're cultured in this chamber here. And then in the next step, you can see this here, you can either transduce them with a viral vector, which is then pumped in here, or you can pump them out and pump them to the left side here. And this is the electroporation device where you can transfer proteins or mRNA to your cell type of interest. And as I mentioned before, I think this really allows you, if you get it up and running, to automate manufacturing protocols and also to produce complex cell therapies on site in your clean room. So coming back to the CAR T cell dilemma, I think what is important to understand is that we talk about cancer patients, and some of these cancer patients don't have a lot of T cells left to produce the CAR T cells. So they're poor donors. So in terms of the quality of the T cells that they can, that they still have in their bodies, or also the quantity of T cells that they have. And what is important to understand, about 10%, for about 10% of cancer patients, no CAR T cells can be produced because, as I said, the quality or the quantity of the original, or let's say of the T cells that you get from the patients are not sufficient. So a lot of people thought about, so what if we move away from patient-derived CAR T cells and produce now CAR T cells from a donor, from an allogeneic donor, from a healthy donor? So meaning that would really simplify the whole manufacturing process, because now you work with a material which is from a healthy donor, and it would also allow you now to stock CAR T cells product in your freezer, if you like, so you would have it available whenever you need them. So it's an off-the-shelf product available right away whenever you have a cancer patient in need of a CAR T cell product. But I think, of course, you also know that T cells will, of course, react very strongly against foreign cells. So if you infuse an allogeneic CAR T cell product into a patient, of course, you will have a lot of alloreactivity, and the CAR T cells will not only attack the cancer cells, but also the patient-owned cells. So we have to eliminate alloreactivity, and there are different ways to do so. And I think we summarized this in a review that we've published a few years ago. So how can you produce such off-the-shelf CAR T cells, or universal CAR T cells, if you like? And of course, one, let's say, straightforward way is to knock out the T cell receptor which recognizes the alloantigens. And in order to knock out the T cell receptor in these CAR T cells, you have to knock out one of the two genes which encodes for the T cell receptor, alpha or beta chain. And the genes are named TRAC or TRBC. So you can design a nuclease to knock out either one of these genes, and you would end up with a universal CAR T cell product, which is non-alloreactive. And you can also further combine this. You can, for instance, also knock out beta-2 microglobulin, so you will not have HLA expression, which will further enhance the safety of your CAR T cells. So we actually said, okay, this is a great starting point. We would like, so we design a nuclease to knock out the alpha chain of the T cell receptor, so the TRAC locus. And you can see this here. This is a schematic. It's on chromosome 14. This is exon 1 of the constant chain of the alpha chain. So we made CRISPR-Cas or tail nucleases to target this gene. And we actually made only a few of them because all of them seem to work quite nicely. You can see this here. This is just a T7 assay to look at the activity of this nuclease. You can see the TALEN knocked out about 85% of the alleles, and our better CRISPR nuclease knocked out about 75% of the alleles. And of course, I mentioned this before, we also looked into safety, into specificity. Back then, we just used software which predicts off-target sites for TALENCE or for CRISPR-Cas nucleases. And then we did targeted amplicon sequencing on these sites. And for the TALEN, we did not find any off-targets. And for the CRISPR nuclease, we found one predicted off-target site here. So we said, okay, the TALEN has higher activity, it has higher specificity, so we moved on to work with the TALEN. Now come back to this machine that I'd introduced before, so the CliniMax Prodigy. And this is now the scheme that we set up to produce these CAR T cells on a clinical scale. And you can see this here. So we have the cleanup, so we enrich for T cells here, we activate the T cells, and we transduce these T cells with a lentiviral vector to express the CAR. And then we electroporated the cells here on this side with the TALEN to knock out T cell receptor gene. And in the end, we cleaned up the final product to deplete all cells which are still TCR positive. So just to start with, as I said, we decided to work with TALEN in that case. So we produced mRNA of these TALENs, and then now optimized the electroporation procedure quite extensively to find, let's say, a program which allows us to have high mRNA transfer efficiency. You can see this here, over 90%, and high cell viability as well. So in the range of 70% is what we reached. And when we now looked at the knockout efficiency, now on the phenotypic level, you can see here TCR expression. You can see that we were able to knock out the T cell receptor in about 40 to 45% of cells. So not as efficient as on the small scale, as I showed you before, but at clinical scale, I think 40% knockout is quite fantastic. So just to do a further evaluation of the product that we produce, as I said, everything was done in collaboration with Miltenyi. So you can see here, this is the lentiviral transduction efficiency, which was in the range of about 45% to 50%. And then we looked at knockout efficiency in 3 runs in total. You can see we reached this about 40% knockout here in average. And as a last step, as I said, we did a TCR alpha beta depletion step, and you can see this is what happens then. You go from 40% knockout to 100% knockout because all TCR expressing cells are now eliminated or depleted. And you can also see that CAR expression remains constant over time, and at the end of our manufacturing process, we still have about 45% of TCR negative cells, which are now CAR positive. And of course, the question is this product safe? Again, we looked at the distribution of CD4 to CD8 cells, and you can see this here. It remained constant over time, about 50 to 50%. We looked whether we have any contaminating cells, especially B cells in the final product or monocytes. And you can also see this here at the end of our manufacturing process. We have mostly T cells, 99%, and we have some very few NK or NKT cells, which are also CD3 positive and TCR negative. But I think this is absolutely tolerable. But as I said, the most important thing is we don't have any B cells left. I think one interesting aspect here, again, was if we do a knockout in these T cells, and if we culture them for 14 days, do we see any, let's say, clonal expansion of a very specific clone, which would be, let's say, which would be indicative of a problem, of course, of genotoxicity. So we looked at the deletions. We only found deletions in that case. In the deletions that we found at the T cell receptor alpha chain locus, and you can see the distribution here. This is run 1 and this is run 2. And what you see is that over time, we did not find any significant outgrowth of a specific clone. I think which was very reassuring for us to see that we don't seem to have a problem with genotoxicity in these cells. And of course, we also looked at the end of our production, the CAR T cells were still active, so we incubated them with target in that case CD19 positive cells, NALM6 or JACO1 cells, and you can see that our CAR T cells were still very efficient in eliminating these cells, and they were as efficient, as you can see here, as cells which have only been transduced without a TCR knockout. So I think all in all, I think we were very happy to see, and that we have now established, let's say, a very straightforward manufacturing protocol to produce gene-edited CAR T cells. So I think I will just summarize this part very quickly. So as I said, we have established a GMP-compliant manufacturing protocol on the CliniMax Prodigy platform. We can show that our final CAR T cell product is 99% pure, and we also can show that these cells maintain potency at the end of the manufacturing process. We did not detect any off-target activity of TALENCE that we used, and I think just as a take-home message as well, I think this protocol that we established now will facilitate the decentralized production of gene-edited CAR T cells in general at many different clinics in the future. I would like to quickly come back to what I said here. No detectable off-target activity, and I mentioned this before. I said we used a prediction tool. And of course, we all know that this is not the best way to look at off-target activity. And of course, the question is, are there better ways to look at off-target activity? Are there better ways to look at specificity of designer nucleases? And I would just like to revisit this issue a little bit in more detail. And I think what I told you before is, of course, you make a designer nuclease to target one specific gene in your genome. So this will be the on-target site on, let's say, this blue chromosome here. But we know that off-target activity can happen at sites which are similar to the on-target site. And let's say this will be on this yellow chromosome here. And of course, if you want to create a knockout, we know that we will have indel mutations at the on-target site, or maybe also larger deletions and inversions. And this is exactly what we would like to see. But we know that this can also happen, of course, at the off-target site. And what we have also learned from quite a few publications which are out there is that if you have concomitant activity at the on-target site and a off-target site, you can also induce translocations. And while indel mutations in genes, or maybe somewhere in the genome, are mostly loss of function mutations, I think we have to be more careful with translocations, because we know from cancer cells that cancer cells often have a lot of translocations, and these translocations are associated with gain of function mutations. So we have to be very careful to make sure that we can characterize these translocations on top of the off-target activity. So if you want to go to the clinic, the regulatory authorities, before you can start your clinical trial, ask you to do some genotoxicity tests. And what people use nowadays, of course, is in silico screening. This is what I showed you before, based on computer algorithms which predict where the off-target sites are in your genome. You can do in vitro assays, so you can cleave genomes in vitro and try to identify where this cleavage happened, or you can use cell-based assays. Usually, these are surrogate assays which allow you to, let's say, specify the specificity of a designer nuclease, but you don't do them normally you don't do them in the clinically relevant cell types. So meaning this will allow you to, let's say, pick your lead candidate, and then with your lead candidate, this will be, of course, your best performing designer nuclease. You have to validate it again in the clinically relevant cell type. And I mentioned before, I think we all know that we cannot be happy with in silico prediction tools. I think they're getting better and better, but still, I think they lack, or they miss a lot of off-target sites. And meaning that you have a lot of false negatives because you don't know that there are other off-target sites somewhere else. So meaning that you have a lack in sensitivity. If you now move to in vitro-based assays, so where you cleave your genome in vitro, of course, this genome is not covered with histones anymore. You will cleave a lot of off-target sites which are maybe not cleaved in the cell. And what I mean by that is false positives. And I put it into parentheses because, of course, these cells are cleaved in vitro, but they're not cleaved in the cell. So that's what I mean by false positives. And which also means you have a lack of specificity because you highly overestimate the number of off-target sites. And cell-based assays, you do it, as I said before, in a surrogate cell line, which is not the patient cells. And again, if you work with patient cells, this can lead to different results. And just to illustrate this a little bit, most people use GuideSeq as a cell-based assay, which is usually performed in a cell line. I took these numbers here from a review which was published by Jin-Soo Kim 2 years ago. And you can see he tried to calculate the validation rate. And he said for a particular nuclease which targets a VEGF-A gene here, GuideSeq was able to identify 22 off-target sites, potential off-target sites. And when he validated these in primary cells, it was about 91% correct. If you now go to an in vitro assay, you can see this here, Circle-seq. Both of these assays come out of Keith Jung's lab in Boston. If you work with Circle-seq, you can find up to 500 potential off-target sites when you use the same nuclease here. And of course, we know that never ever these 500 sites will be cleaved in a cell, and the validation rate was 30%. So meaning you, as I said before, you highly overestimate the number of off-targets in cells. So we said, okay, I think we need a better assay to look into this. And we said we would like to develop an assay which not only allows us to determine off-target activity, but also at the same time to look into translocations. And it should also be an assay which can be performed directly in the clinically relevant cell type. That means in the cell that you would like to transfer to the patient. So this is how we set up this assay, and we did it in CD34 hematopoietic stem cells. And then we nucleoaffected these cells 2 days later after taking the cells into culture with CRISPR-Cas nucleases. This was done in the form of protein here, or with TALENCE, again in the form of mRNA. And you can see we get typically indels in the range of 40 to 95%, depending on the nuclease. We culture the cells for 1 to 14 days, then we extract the genomic DNA, and you can see we just need very few or very little genomic DNA. We can live with about 500 nanograms of genomic DNA for one test, which corresponds to about 75,000 cells. I think this is important later on if you want to use it in the clinic as well. And as I said, once you've done your sequence analysis, you go to the bioinformatician, and hopefully he can help you with the data analysis. So I think we needed a little trick to get it to work, and I would like to introduce you to this little trick that we used. Let's assume again, we have the blue chromosome with our on-target site, and we have a yellow chromosome which contains an off-target site. And you will have cleavage in the cells, and as I said, in very few cells, and we estimated it will be less than 1%, you will have translocation as well. And this is what you're looking for. You don't look for this, you look for this. So you fragment the genomic DNA, you do end repair, then you ligate your linkers to it, and here depicted in red. And then you do a first PCR to enrich now for the translocations event. And in order to really enrich for translocations event, this is now the little trick that we use. And this is what we call the addition of decoy primers. And the decoy primers, you can see them here. So again, blue would be our on-target site. We have a bait primer to the on-target site. And we have a prey primer, which binds here to the linker. So this is the red primer here. And now to detarget from the on-target site, we add these decoy primers. And the decoy primers, of course, bind to the on-target site, but on the other side of the break in reference to the bait primer. You can see this here. You can see what happens. As soon as you add these decoy primers here, you will have not a large PCR product between the prey and the bait prime, but we have two shorter PCR products. And you can see this is depicted here. You will not find any full-length PCR product anymore. So I think this is a very efficient tool, a very simple, but a very efficient tool to detarget from the on-target side. And you can see, of course, the decoy primers will not bind to a translocation side because this sequence here is different from this. And meaning you now shift the balance from the on-target side to this translocation event here. You do a second PCR now to amplify, to further amplify, and as I mentioned before, you will not have an amplification product here because this product is interrupted here. So you can nicely enrich for translocations. And the third PCR, you add the barcode for sequencing later on. And as I said, this little trick really helped us to get to a sensitivity, which is 1 event in 10,000 cells. And just to remind you, if you do target amplicon sequencing for looking for indel mutations, for instance, your sensitivity is in the range of 1 in 1,000 cells. So you can see this is already much better than what you just get by simple sequencing. So this is the work of our bioinformaticians. I don't say that I understand everything in detail, but I think what is important for you guys to understand is that we can classify now the events that we identify through CAST-Seq into 3 classes. So one class that we identify is off-target mediated translocation. This is what I introduced before. We found a novel class of aberrations in these cells, which we call homology-mediated translocations. And I will get back to this point later on. And because our assay is so sensitive, you also find natural break sites. So if you have an on-target cleavage event and you have a natural break at the same time, you will have translocation there as well. And this is what we find as well. But of course, they are not relevant in this context because they just happen randomly. So just to get to the point and to be more specific, and I think also to introduce the technology and to visualize how it actually works. So this is raw data here that we get back from the CAST-Seq run. We look now here at the genome. This is the CCR5 target site. And in close proximity to CCR5, we have a second gene, which is called CCR2. And we already know that it shares high homology to the on-target site here. You can see this is the number of reads that we get. So this is cut off here, the raw data. And you see this is the number of reads that we get. And you see the peak here, close to the on-target site. And you see a second peak here. So what you can see here, we get a lot of noise around the on-target site. This is indicative of large deletion and inversions at the on-target site that we get after introducing a double-strand break. You can see this peak here, which inserts a large deletion of about 15 kb. And you will see later on that we don't have cleavage at this point here. And I will show you later on that this is mainly based on homology between CCR2 and CCR5. And just as another example here, which occurs very infrequently, but this will be a true off-target site here, which then can lead, for instance, to non-reciprocal translocations. You can see here, you have, for instance, one chromosome without the centromere now, and the other chromosome would have 2 centromeres. And of course, these are non-viable cells, so you will lose them over time. So I not just talked about chromosome 3, but of course, the off-target site can also happen on a different chromosome. And in that case, you will have translocations, so true translocations between the blue chromosome and the yellow chromosome. And what I would like to point out as well is that all of these events here cannot be predicted by any existing assay so far. So I think it's very important to have this novel assay at hand to also get a more complete picture of off-target effects. So this is when you clean up the data, it looks like this. So coming back to this CCR5 targeting CRISPR-Cas nuclease, you can see this here. This is the lineup of our chromosomes here now in a circle, so from 1 to 22 and then XY. And you can see here, this is a blow up of chromosome 3 with our target site, and this is CCR2 close by. And now you can see the red lines here indicate true translocations. You can see this here. This is a translocation from chromosome 3 to 13, a translocation from 3 to 19, a translocation from 3 to 22, and so on. And you can see this aberration between CCR2 and CCR5 here indicated as well. So when you look at the breakpoints, so where do the translocations happen, you actually can see that at the breakpoints for these red lines here, we actually find sites which are similar to the on-target site. When you look at the sequence here, you can see there's high similarity to the on-target site. When we look at this site here on CCR2, you can see there is high similarity, but the PAM is not there. So this also explains why we don't see cleavage. So we did now NGS sequencing and to look at indels at these predicted off-target sites, if you like. And what we can confirm by NGS sequencing is we have high on-target activity. You can see about 90% here. As I mentioned before, we don't see cleavage at the CCR2 site, so nothing over background here. And we have indel mutations at the predicted off-target sites where our translocations have happened between 1% and about 15% here. So the next question we ask, can we improve the situation if we use now high fidelity Cas9 protein, for instance, something you can buy from IDT? And the answer to the question is yes, you can improve the situation. You can see this here. When we now look at off-target activity, you can see the black bars here. We can reduce off-target activity of 2 of these 4 off-target activity sites. You can see it goes now down to background here. And when we now look at the translocation events, and these are now the number of hits, translocations, if you like, that we identify, you can see this here again. For this site here, we don't see translocations anymore, and for this site here, we don't see translocations anymore. So meaning, if you don't have off-target activity, or if you can get rid of off-target activity, you can also get rid of translocations. And as I mentioned before, of course, the situation at CCR2 did not change because we didn't have cleavage before, but the number of translocations or aberrations in that case, I should say, stays high because this is independent of off-target activity. So this is the summary here from this slide. So high-fi Cas9 proteins can reduce translocation frequency, but it cannot prevent these homology-mediated aberrations. Is it possible to, let's say, generate highly active but also highly specific CRISPR-Cas nuclease? And the answer to that question is yes, it is possible. So we made another nuclease to target CCR5, and you can see this here. We see a lot of noise around the on-target side, but we don't see any translocations. And the same was true for this CRISPR-Cas nuclease to target the Fanconi F gene. You can see some noise around the on-target side, but no additional translocations. So it is possible to produce highly active and highly specific CRISPR-Cas nucleases. We also made a TALEN to target the hemoglobin beta locus, HBB. And this is a similar situation that you find here. Close to HBB is the hemoglobin delta gene. And again, these 2 genes are very similar to each other. And again, you can see this here. We find a lot of aberrations between the 2 sites. And you can see this here as well. While we have few translocations, so it's not a perfect TALEN in that case, we have a lot of hits between the 2 genes. So again, homology-mediated translocation is the problem here. And one of the reviewers of our paper asked, is this really homology-driven translocations? And a simple assay and a simple way to prove this was to add now an inhibitor of a key protein in the HR pathway, which is RAD51. So we used the RAD51 inhibitor. We looked at translocation frequency, which was off-target mediated, and you can see here, these brown bars. We don't see any difference. But when we look now at homology-mediated translocation, you can see we have a significant decrease in the number of translocation frequency for CCR5, CCR2, as well as for HBB and HBD. So it really makes the point that this is homology-driven and not off-target driven. Another reviewer wanted to know if we now have translocations, and this is an example for a RAG1-targeting CRISPR-Cas nuclease. If we have translocations, as indicated here, do we also see translocations between 2 off-target sites? So we set up a PCR because our assay is always anchored at the on-target site, so it cannot detect these translocations here. But we will predict, and it was also published, that this happens. And so we did a simple PCR to see whether we have translocations between chromosome 4 and 14. And our simple PCR said, yes, there are translocations. And just to make this picture complete, I think it's always important to keep in mind, if you have high off-target activity, you will always find translocations between two off-target sites as well. The next question is CAST-Seq quantitative? I think this would, of course, be really cool, because then you know exactly how many translocations you have in your cell population. So we looked into this in more detail. So these are the raw data that we took here from the CCR5 as well as from the RAG1-targeting nuclease. And you can see we line them up. So we line up the number of hits that we get in our CAST-Seq analysis, and then did a ddPCR, so a digital droplet PCR, to quantify the number of translocations. And you can see that the numbers line up very nicely. So there is a nice correlation between the CAST-Seq hits and the number of translocations events in our population. So there is about a 1 to 10 ratio, meaning that one hit in our CAST-Seq analysis corresponds to about 10 true translocations in the cell population. And this now really allows us to, let's say, add numbers to the events that we see. So it's not only qualitative, but it's also quantitative. And you can see, for instance, this aberration between CCR5 and CCR2 happens in 1.6% of alleles. These translocations here happening in about 0.5% of alleles. And you can see here, this is the limit of detection here in about 1 in 10,000 cells. And this now brings me to my last data slide. And I mentioned this before, that we have a lot of noise around the on-target site. And we wanted to quantify this noise, if you like. So how many large deletions or inversions do we have at the on-target site? So we set up, again, a ddPCR assay, which now amplifies the cleavage site. You can see here, this is called the HMTs. So this is our cleavage site. These are the two primary binding sites here. So we amplify the cleavage site here. And we have two flanking amplicons, which bind to the same chromosome a little bit further away from the cleavage site to allow us to detect large deletion events. And maybe just focus on this part here of the table. So if we have a large deletion, we would expect that the H amplicon will be negative, as well as the flanking amplicons, or at least one of the flanking amplicons will be negative as well, because the large deletion will delete the primary binding sites. If you have a true translocation event, the H amplicon will be negative, because one of the two primary binding sites will be gone, but the flanking amplicons are still there, because independent where the translocations happens, the amplicon is still possible to be amplified. So, and this now really allowed us to look into more details. I don't want to go into details of this copy number variation detection that we made, but I think the bottom line is this here. So if you just apply NGS sequencing or a T7 assay, what you will get when you look at this site here is you will see, okay, I have about 80% indel activity or 80% knockout. You miss all the large deletion events, and you miss all the translocations events. And for this very particular nuclease here, so the CCR5 targeting nuclease, we now can quantify that we have about 10% or 15% of large deletions. And we have about 5% other aberrations, which means chromosomal translocations or this, other aberrations that I showed you before. So meaning you have additional 20% of events which you miss by just sequencing or T7 assay. And I think this is a very important number to keep in mind here. So just to summarize what I told you that we have developed a novel assay, which we say it's a quantitative diagnostic tool. It's not a prediction assay. It really quantifies the translocation events that you find in a cell population. and in the clinically relevant cell type that you then can transfer to the patient. And we find that up to 5% of cells can have translocations, off-target mediated translocations, and these off-target mediated translocations can be reduced by hi-fi CRISPR-Cas systems, and they can also be reduced by having a good choice of a target site. I think this is very important to keep in mind. I showed you FANC-F, for instance. This was a normal Cas9, and nonetheless, we did not find any translocation. So a good choice of target site is very crucial here. I showed you in my last slide that we have on-target aberrations, which have been overlooked so far in 10 to 20% of cells. This was independent of the target site, independent of the platform that we use, so TALENCE as well as CRISPR-Cas nucleases. And I think this is just a number that is important to keep in mind. We see now these non-reciprocal translocations. I did not go into detail here, but they're lost over time. And we detected a novel type of translocations, which are these homology-mediated translocations. If you have a neighboring gene which is highly homologous to your target gene, this can be very frequent, as you can see this here, 1.6% in the case of CCR2, CCR5. So this is intrachromosomal, but we also detected them interchromosomally between two different chromosomes, although they seem to be very rare in that case. So what I would like you to take home is, I think this is a very nice clinical risk assessment tool. It allows you to identify your lead candidate. It allows you to do a proper risk assessment before you go to a clinical trial. And then in a second stage, once you started a clinical trial, it allows you to do to continue with your risk assessment in longitudinal studies, so you can follow these patients over many years and see whether there is a change in the population in these cells. And I think this will be important then for marketing authorization of your product if you are a big pharma company. So I think it is important to look at off-target activity, but I think it's equally important to look at translocations, and you can complement these two assays, for instance, with cancer panel sequencing. A good target to look at is, of course, the p53 gene, because we know that the p53 mutations can allow you to get better gene editing in stem cells, for instance. I think it's very important to make sure that you don't have mutations in p53. Let me thank the people who did the work. In yellow, you see the people who did the manufacturing part. This was mainly Tatiana Cornu's group here, GMP development with her team, with the help of Miltenyi, as I mentioned before. And the CAST-Seq assay was developed by Jando and then taken over by Julia. And I think it was only possible to get it to work because we had great support and great input from our biomedical bioinformatics team, from Melanie and from Geoffroy. So we'll stop here, and I'm more than happy to take on some questions.
Thank you very much, Toni, for this wonderful talk. Really excited. And I just would like to invite our audience to type in the questions so that I can read them to Toni. And I think I saw already the first. Yes. So Maya from Charité asked, how do you monitor off-target toxicity in vivo in patients?
So I think what we would do in vivo is, of course, you try to get a biopsy from the patient. Let's say if you have in vivo genome editing in the liver, you try to get a liver biopsy, and then you can do your analysis on this liver biopsy. Or maybe you have in vivo, well, I think you can do it with any tissue, with muscle cells as well. And I think that's why it is important that we only need very few cells to do the analysis. And I think this is probably how it will be done in the future, just to follow these patients over time and to see whether we have any clonal expansion of cells also in the liver. This might be quite deleterious.
I have one question, Toni. You say you have this fantastic diagnostic tool, and it really looks great. What is your magic number when it is safe to move forward? In particular, as you said, you would use it first, of course, to set up this the whole study and then also for monitoring. So what would be a magic number there?
Yeah. Well, I think I've shown you that it is possible to edit the genome without detectable translocations. So I think the magic number would be 0 translocations. But I think what you cannot get rid of is, of course, the on-target aberrations. I think we have to live with them. But I think on the other hand, maybe there is no magic number. I think you can have translocations which are not deleterious. I think what is important to do in that case is really to do a proper clinical risk assessment, meaning we know where the translocation breakpoints are, and we know now what genes are next to each other. And we actually looked into this as well. And we now see that is a translocation, or let's say is an oncogene now in close proximity to another promoter. And of course, if you find something like this, I think this would be a complete no-go. Yeah.
Thanks. Michel Antino asked, long-range PCR and long-read sequencing are needed to detect all undesirable on-target and off-target mutations from CRISPR-Cas. Do you do this? No, we haven't done any long-range PCR, but I think this is, I think it is important to do as well. I think right now we just base everything that I said before is based on our CAST-Seq analysis. But I think with long-range PCR, you will get an even better picture of what is going on at the on-target side. This is absolutely true. Yeah. Razzi Moniesi, a postdoc researcher from University Clinic Würzburg, asked, have you ever tested the off-target activity and translocation events when CRISPR-Cas is provided as RNP as compared to other formats? I think we have compared RNA-based delivery of CRISPR-Cas versus RNP-based delivery of CRISPR-Cas. I think we did not see that many differences, I have to say, because I think both of them are short-lived expression of the CRISPR-Cas nuclease. I think what other people have shown before, if you have plasmid-based expression of CRISPR-Cas nuclease, I think there you will have much more off-target activity because you have a, let's say, high peak expression and long duration of expression. I think, of course, this really increases your risk of inducing translocation. So I would say the concentration of CRISPR-Cas nuclease is important, as well as the duration of expression. And if you can, let's say, you find the right amount that is needed to do the on-target job and as short as possible, I think then you're on the safer side. Yeah. I don't know whom is asking this, but first of all, there's a thank you, Toni, for this talk. And then can you please comment on the untreated or background observed with CAST-Seq and how it varies across cell types? The background, I'm not sure what it is. Natural break sites, whether this is the background that this person refers to. I think we always find natural break sites. As I said, I think the assay is extremely sensitive, and that's why you find them. But I think if you repeat the assay, if you do a biological replicate, you don't find the same natural break sites anymore. And I think this is how we also can validate these are natural break sites and not, let's say, true off-target events. I think this is important to understand. But I think we work with primary hematopoietic stem cells. I think if you work with cancer cells, you will even have more natural break sites. I don't know. We haven't done the experiment. I think this is just a prediction that I'm allowed to have, I guess. I mean, this brings me to another question, of course. So if you compare the different cell types, and can you somehow let us know what you have learned in all the years with regard to primary cells, specific cell types, cancer cells, and so on, with the risk of off-target? And yes, of course, translocation you also already discussed, but just with regard to safety and the use of design nucleases there. Yeah, I think the chromatin configuration has a major impact on off-target activity. And we haven't done it yet. I think now we start to compare
T cells versus blood stem cells, and maybe a third cell type to see whether we find differences in translocations. I don't have an answer to this yet, but I would predict, yes, maybe in some cases, we will see differences in translocation frequencies, or maybe in translocations at all. If you do these assays, and this is another prediction because we haven't done the experiment, but if you do this assay in cell lines, we know that in many cell lines, the chromatin is open. If you work with HEK cells, for instance, the chromatin is completely open, and you will have many more off-target activity, much more off-target activity, and therefore probably also many more translocations. Again, this is a prediction, but I think this is, I would bet on this quite a bit of money.
So the next question is from George Dixon, and he's asking how reproducible is the outcome batch to batch? Where's the cutoff to prohibit a CT application?
As what application?
Clinical trial application.
Clinical trial application. Okay, yeah, okay. So we see reproducibility. I think we see great reproducibility for, let's say, frequent off-target events. Translocations. If the translocation is close to our limit of detection, like this 0.1%, we don't detect it all the time. So if you repeat the assay over and over again, we sometimes detect it, we sometimes don't detect it because it's really close to the limit of detection. As I think for clinical trial authorization, this is what we discussed before, I think maybe there's no magic number. In the ideal world, you don't have any translocations. If you have translocations, because maybe you cannot avoid them, I think I would do a risk assessment based on how the translocation now looks like. What is in close proximity now? If you have a strong promoter close to an oncogene, this is a no-go. Yeah. But I think we will probably learn more and more. I think there is not a lot of information out there. I think what we have available right now is just based on sequencing of tumor cells. And of course, in tumor cells, you already have a high degree of evolution of these tumors. And what we look at is the end product. And we, of course, these tumor cells will have translocations where exactly this happens. So you have a strong promoter, which drives now a proto-oncogene. I think we need more information to have a more complete database. And I think the more complete the database is, the better our predictions will be, whether this is potentially dangerous in the future or not.
Juan Rodriguez Mados from CIMA, University of Navarra, is asking, I would like to know if IGUIDE assays is complementary to CAST-Seq and if both techniques should be performed to characterize the safety.
Yes, yes. I think CAST-Seq, I hope I could convince you, can detect off-targets based on the translocations that we identify. And when we look at our translocation breakpoints and we look at the indels, we find indels at these points. But we also know in a few instances, for instance, we found translocations and we did not find indels. Okay, meaning that maybe this was homology mediated, we don't know, but we didn't find homology. So something, meaning that with off-target activity, sometimes you don't detect them because they're under the limit of detection of NGS. Nonetheless, we see translocations, and vice versa. We find off-target activity in 2 or 3 cases, and we don't find translocations. So maybe the translocation led to a product which was not viable to the cell. So meaning we have a natural selection in the cells, of course, as well. So the short answer is yes, do both. Do off-target assays using a GUIDE-seq or whatever, and combine it with CAST-Seq to look at translocations. And in an ideal world, you would see 90–95% overlap.
Thanks. So I'm just quickly have a look into the chat if we have another question. It seems not to be the case, but I'm sure you would be available. If anybody has further questions, he will or she will just shoot you an email, I assume. And I really would like to thank you for this great talk and our audience for the discussion. And if you give me my last slide, I really would like to invite you all back to the third seminar of this little series, which would be next Wednesday. And we will then have a talk by Laura Sepp Lorenzino. And by this, I really would like to say goodbye and have a nice week. Bye-bye.